adventures with mussels. Actually, nicely extending on what Nigel has touched upon in the beginning of his talk. So using individual-based approaches, agent-based approaches in ecological systems, and also the patterns that you can form and can explain with individual-based approaches. So, so I think most of you know that generally there are two approaches in science. If it comes to modeling, you can use the Eulerian approach, like you have a landscape, well, a grass field, and you cut it in little pieces, and you model it as little collections, little ecosystems on their own with spatial expansion between them. That works because if you have a grass field, there are so many strands of grass in little parts of your chessboard that's no problem. If you then put like 20 cows on that grass field, and you think like, well, let's see how many, and then suddenly you fit, well, there's only like 0.1 cow fitting on my one square. So then it doesn't work anymore. So that's, of course, where Lagrangian approaches are working much better and are useful. And that's an approach which, of course, also found its way in ecology. Because, of course, yeah, if it comes to organisms, particularly if you look at the way that biologists approach organisms and also evolutionary process, for instance, like you appreciate very much that there's all this biodiversity, but also many species are all different. Individuals are all different. One is a bit bigger, the other is a bit smaller, and evolution works on that. And so, to appreciate the diverse nature of organisms, individual-based approaches seemed very logical. And that has developed, I think, from the 80s, 90s. Volker Grimm was a big name in that world. And he made, at some point, he got a request to make a book for the Princeton monographs. And he basically wrote the Bible on how you make individual-based models. And that got something going. It's really nice to mention that. So he basically says, like, well, if you make it, we should all make individual-based models. Ecologists, stop using grid-based models. Don't do that anymore. It's not appropriate. Shouldn't do that. Because we need to include details about each individual life cycle. Must be present in these models, including where it's born, how it moves, where it dies, as events, and so on. And so the dynamics of resources used by individuals must be explicitly represented. So, sorry, no carrying capacity anymore. You need to explicitly take the resources in account. He didn't talk about what you do with phosphate as a resource, with millions and millions and millions and millions and billions of individual atoms, but and so on. And so use integers and not real numbers for populations, and discrete events for things that happen. If you're predated, that's an indiscrete event. That's not a flux. And variability between the individuals must be allowed and must exist between individuals of the same age even. So, well, you can imagine what happened if you write a book like that. You get people going. And so a comment on that came from Joan Roughgarden, who is a theoretical ecologist. And she said, like, well, why would you come up with all these unnecessary requirements? How to not to ruin a good thing was her response in a sort of a criticism paper. And she said, like, many examples exist about really important IBMs that do not fulfill the Grimm-Railsback criteria. So, highly cited papers, not good enough, apparently. That's interesting. So individual-oriented models, or even ODEs, are often sufficient to reach the goal. If you want to explain something simple, you don't need necessarily build something complicated. And IBMs are not superior, was her argument, but inferior. There are all sorts of possibilities. All these techniques that Nigel has been talking about are much harder to implement, if at all possible, using IBMs. So it was a lot of feedback. And of course, well, if we start looking at the differences between these sorts of models, then of course ODEs function differently. They are working on something that is an amount per spatial unit, while an IBM looks at an individual. The dynamics are determined by fluxes, which is sometimes, of course, a very logical thing to do. The dynamics of individual movement are important in IBMs. Particularly movement, but also proximities, events, and individual changes. Parameters are determined on the population scale, which sometimes actually makes it quite hard. Like I told you yesterday, like there's some C times some A, algae, times some M, mussels, determine how much algae is consumed. How do you ever measure the C? It's very complicated. It's much easier, as I will explain myself, to actually to measure it on individuals. How much did that individual eat? Much easier to investigate. And random events are, of course, very important in IBMs. You move. How an organism makes a decision to move that way, and the next time it decides to move that way. Of course, that's not a random event, but it's approximated by assuming that it's just a sort of random chance whether it moves right or left. And of course, that's much less the case so far in OD and PDE models, although Nigel is doing his best to improve that. Okay, so advantages, of course, is that of IBMs is that it addresses variation between individual organisms, which, of course, particularly for instance in evolutionary context, is actually, I think, a good thing to do. And of course, there's no aggregation artifacts, but disadvantages are that computational times can be very big. They're getting bigger and bigger. You saw how nicely it was flashing across the screen on the computation that Nigel showed. But the thing is also, it's often sort of a black box. I mean, you saw the white sharks and the black fish, or the other way around, and you actually don't know what's happening there. It's more difficult to quantify, particularly if something happens that's more than what you put in. And that last part is also something that I will talk about. It's more difficult to then link back to in the theories about differential equation-based theory. And so how do you make this comparison? Everybody making an individual-based model has a danger. I will give an example myself about sitting on a little island, which is, you make your model, it's different from that model, different from that model, different from that model. You don't know precisely how it is different. How do you make a sort of a uniform setup for individual-based models so that that's better possible? And well, it's strongly debated. It's just a little summary. And so I would actually advise you to be careful with using IBMs unless you really clearly see the advantage for that. And only individual elements are crucial. And not just because they are nice to use. I mean, there's always appeal, particularly about net logo. We see a sheep, and there's a little wolf. It looks cute, but we're not in Disneyland. And so I think we need to address that seriously. Of course, for educational purposes, these things are really nice. And also actually to show the difference between these two approaches. Okay, so this is a little introduction to the whole idea of individual-based models. I actually had a colleague, it's called Wolf Moy. He was very much in the team with Wolf Krim, and he was also telling me like, oh, yeah, you have to make individual-based models. And I also think like, nah, come on, be serious. I'm not going to do that. But at some point, actually, I changed my mind. And I'm going to tell that story. And that's a story that deals with mussels, as you can see here. And pattern formation in mussels, but then on a different scale, much smaller. And the reason for me to come here is actually an interesting story. So this is much more a story about how I did my research in the past. I think it's nice to tell. Also, it really shows you what sort of decisions you make when you're doing your research and how that sometimes can lead to completely unexpected results. And so it starts with this actually. I mean, I was studying mussel beds and theorizing and making a model and then pressing run and presenting it, and it's like, us complexity scientists, we understand it all, and of course never presenting any evidence for what happens, which is typically happening. The typical person working on arid systems, most of them have never even been there, and they make a model and make sure that the colors are similar, and say that they understand how it works, and of course I can't blame them because I was doing exactly the same. But other people were criticizing me for that and saying, well, you can test that and you can test it. Why don't you do that? You're going the easy way. And so, yeah, at some point, I was always saying, oh, next year. But that you can only do this so many times. And so at some point I thought, okay, let's do that. Let's go. And actually, I had some colleagues in Bangor, which I was communicating with. So that's in England. Big error, that's in Wales. To the west of England, and here you have the top part of Wales, and here's the Isle of Anglesey, and there's a nice strait in between, and it's a very nice place. It looks like this. That's the advantage when you're not a theoretician. You can sometimes go to these very beautiful places you're being paid for, and do research. And this is how it looked like. This was when I came there actually, and it's just a random spot with a nice view on the city of Bangor. But before I arrived, they sent me these photographs of these mussel beds. You've seen them yesterday also. And of course, they look gorgeous. They look like mathematics. This is almost not the real world. I mean, so beautifully, smoothly curved. And they want to say they don't look like this. They're much more perturbed, and edges are off, patches are gone, and all these things. But here they're beautiful. But then 3 weeks before I applied for a bit of money, a few €1,000 to go there, and so do some research. And then I arrived there. It's in 2003, so really some time ago. And she said, like, "Oh, we have a problem. We have a problem. The fishermen fished them all away. It's all gone." And I had made plans of measuring algal depletion and doing this and doing that and so on. And nothing actually worked anymore. And so this is what was left. And yeah, it doesn't look as beautiful, isn't it? But so yeah, I was there. You can sit in the pub for 10 days and not do anything. But I felt bad about that because, well, the taxpayer is paying. And then I thought, like, well, if you have a good look there, it actually looks pretty interesting on a small scale. You have these small scale patterns in this system. And that's a really interesting thing. Maybe we should look at that. This pattern, it looks like a tiger bush almost, but just on a square meter rather than on a square kilometer. And so, yeah, this looks really nice, so maybe work on this. And then there was a sort of local, well, he was an assistant, I think in some way hired by the local university, but he called himself a fisherman. And he came from the local community, and he said, you don't have to study this. This is like, us fishermen, we know this. This is just common knowledge here. You take mussels, you put them in a bucket, they make these patterns straight in front of you. And I thought, like, that's cool. That sounds nice. You can just make these, you can just experimentally make a pattern, and then do things with it, investigate. Actually, you only go there, in Africa, of course, there are hundreds of years old. The mussel beds here, you don't see them developing. But if it's possible to just have them developing on a short time scale, that would be really cool. And so I thought that was a great idea. And also because if you looked at the literature and looked at what would be the best example for experimental evidence of pattern formation in any ecosystem, it actually came from this. It was spatial pattern formation in ant colonies, where Guy Therolas, a person from the University of Toulouse, he made a setup like this. It's a petri dish, but a big one, with a hole in the middle. And around the ring there are dead corpses all corpses are dead, of course, but of ants, dead ants he had laid out. And then he let live ants out from the hole. And ants have a tendency that if there's a dead one somewhere, they take it, they pick it, and they put it next to another dead one, because they want to make piles of, get them out of the way, make a pile of them. That is how ant life works. And then they make a sort of a pattern on the edge, where you see a sort of a regular pattern developing in these ants. And he also mentioned like Turing and morphogenesis, and so the whole theory of, nowadays, of course, we would think a little bit different about that, but in 2002, this was a bit of the setting and the best sort of experimental example that was available. So I thought, I can do better. That's cool. This is nice. This is a paper in PNES, and I can do better. So we did all sorts of things there. We tested all movements, but I actually went home after 10 days because the experimental stuff would need a lab. And so I went home, took myself a container. I'm a sort of a shoestring experimentalist. Yeah, these people that make these huge, fancy, very complicated experiments. That I don't like that. I want to have, just give me a container, a bunch of tubes, and hook my computer to it on a webcam, and you can sort of do cool stuff. And then you can do it after like a few days rather than having to design a whole experiment. And this is how it looks like. So you feed them a little bit of algae from this bucket as food. And then I thought, like, okay, experimental pattern formation. Turing had this symmetry breaking instability. So you make it homogeneous as you can, and you add a little perturbance, and you see how this then works. Well, making perturbance is not such a big deal, difficult, because the muscles are constantly doing things, so they perturb themselves. And then I will show you a movie, and this movie is during, I think, about a day. I'm not sure if it's a whole day or the first hours, but it's a certain time period. Every second, I think, is about 30 minutes, I think, 25 minutes maybe. So it's one snapshot per minute. And this is how it looks like. So you see very quickly, the fisherman was perfectly right. They made these patterns straight in front of you. So I thought, like, oh, this is incredibly cool. You can then change things. You can change the conditions and see how this alters the pattern. That's very difficult on an ecosystem scale. Let's have less muscles, and then this is how they move. So I was completely enthusiastic, and I phoned or emailed, I don't remember anymore, my colleague Max Rietkerk, who was working on these arid systems. I thought, I have an experimental system of pattern formation. Isn't it cool? And he said, like, well, I don't know. Why is this pattern formation? This is aggregation. This is aggregate. I don't see. Why would it be self-organization? I actually asked him, but this is experimental system for self-organization. This was how I mentioned it. So he wasn't convinced. And I took it as a bit of a challenge. I think actually he was just jealous. But at least I tell myself in the mirror. And so I thought, like, okay, let's study that. And then let's see whether we can measure what is happening, put that into a model, and then show that pattern formation can be explained by the interactions between the individual organisms, because that's the idea behind self-organization. And of course, what I had was 1,440 pictures for each day, and I typically run it for a day. And so, yeah, how do you study that? You can, of course, this is, I think, a few thousand, a few hundred, I don't remember anymore how many muscles there were. You cannot track every individual muscle, how it moves about. Of course, you want to track a few, but how do I then know how? I mean, it's a process of aggregation. So the other muscles move closer, and as they have more neighbors, they become more happy. Maybe they make bissel threads, and you know the story. And so, how do you do this? Well, I thought, like, well, I cannot count them all. So I will just look at, for every muscle that I track, I look at its neighborhood, how much of the neighborhood is covered by other muscles. So it becomes a value between 0 and 1. And that basically I looked at whether I could explain the movement rate of the muscles based on the cover of other muscles in the neighborhood. And of course, you exclude the muscle itself, so that's why there's a little blue hole in the middle. And so this is what the relation looked like. It's a bit complex. It doesn't look like it follows any central limit theory. So it's noisy, it seems to go down, and we fitted all sorts of distributions, and it's basically an experimental distribution, meaning most of the time the muscle sits still, and sometimes it makes a step, but the bigger the step, the smaller the chance of it happening. And of course, this step is something happening in a minute. It's not a real muscle step, it's a movement that happens within a minute. And so we fitted that with an exponential distribution, and build a model for that. So you use n individual muscles, and the speed decreases, or the step size decreases with local density of muscles. And the angle is random. It, of course, isn't really random, but just as an approximation. And movement speed follows an exponential distribution. And so we put this into a model, and this is how it looks like. And of course, with a little bit of perturbation, very small. And this is what it did. So, okay. Would we be happy with this? Is this a pattern? Who thinks this is a pattern? Nobody. Who thinks this is not a pattern? Okay, a few of them. Of course, it's always a pattern, but this is a sort of a pattern that we, yeah? Yeah, that's a good point. It is not homogeneous, so that means it is sort of a pattern. But it's not the pattern that I was looking for. Let's put it like that. And if you actually let it go on for a very long time, then it actually aggregates and aggregates and aggregates and aggregates and so on. And so I wasn't happy with this. This didn't look like how a muscle bed looked like. So I thought, like, so in the model, they make these very, very big patches. In the real world, they don't do that. But I don't know how they would behave in very, very big patches because I never have them in very, very big patches. So let's make big patches. So I made patches of 2 muscles, of 8 muscles, of 32 muscles, and of 128 muscles, and 128 muscles is too big. And the 32 would be, I guess, the right amount of muscles if you look at these clumps of muscles. And so and I looked at how they then moved, and what you see is that they move a lot at a cluster size of 2. They didn't move that much at 8 and 32, significantly less, and it went up again at 128, at least compared to the 32. So you see a sort of a parabolic relation, roughly, at least a bimodal one. And you also see that if you look at these large patches, it's not that muscles move out of it, but they move more inside. They start to look for a better location. As a consequence of that, these patches disintegrate and become elongated. So actually, you could argue that the local density of muscles within any point becomes less. And so let's put this into the model. And well, yeah. So we looked at this as a two-scale effect. I'm going a bit too fast. And so I looked at the data, and so I started to make different rings with different sizes and see whether I could. I made a whole range of sizes and look for, well, which size would be most relevant. And I used a statistical technique, basically stepwise regression, where I offered every ring size as a potential explanator for the movement of the muscles. And then basically this came out. And you think like, well, there's no data. That's because it's just one big cloud of points. You don't see anything in it. But if you would start fitting, then you see that the smallest ring is the best explanator. If you remove the variance explained by the smallest ring and look for the residuals, then the residuals are best explained by a cover I think was about 7 centimeters distance. And so you have apparently this two-scale effect. They like to be close, but the movement goes down if you have more muscles in your direct neighborhood. But if at a larger scale the cluster becomes bigger, then they move out again. They move more again. And so this I put into the model. And then this is how it looks like. And then if you run the model, it does this. So it works much better. So I thought like, well, I think I have... I went to Max and said, does this convince you? And he was actually quite enthusiastic about it. So that is how we... and in the end... so the conclusion is the interaction between individuals explain pattern formation here. So aggregation for safety and dispersion. Actually, we tested for that. We thought like, when in these very big clumps, why are they moving out? Well, we thought like, maybe they want more food. And so what we did is, they don't have enough food. And what we did is we provided them with food. And also we provided them, we made a little tube underneath the plastic sheet. We supplied them with algae. And you could see then, if you would do that, that the movement rate of the muscles dramatically decreased. Of course, we thought like, well, another possibility is that it's nothing to do with food, but with waste products. So also we provided water without algae. So we flushed them. And the flushing had no effect. And so in this way, we could establish that it was sort of food limitation that would drive them out of these very big patches. Although initially, I didn't have that much food in there. So it's not that they necessarily wait for the food limitation to occur, but if you then provide them with food, then they don't mind anymore. So it's actually a pretty complex thing. And so my conclusion was that, well, that's not just aggregation, self-organization. So an example of experimental example for self-organization. And of course, nice is that you have a model that is completely parameterized. You can do that with individual-based model. It's completely parameterized based on the observations that we have from this, from our recordings. And so we thought, like, well, if you can send this ant stuff to PNES, then this can maybe go a little higher. So we sent it first to Nature, and we got it back very quickly. They immediately pressed the auto reject button. And then we sent it to Science, and Science was it's actually a nice story. Science was positively, but one of the reviewers said, like, this has already been done. And so they pointed me to somebody at my own institute who had done that and published that in 1942. It made containers, put mussels underneath it, put glass on it, transparent paper, and had traced all the mussels there. So I guess the reviewer was somebody who knew my own institute very well. And that person was, I think, had already died. But there was, I think, just a funny remark from his side, because the topic was completely different. And and so we landed the paper in Science, my only first authorship. And so I sort of show off with my paper in Science, but you should think of how did this come about. I went with a whole plan to Bangor. I would have made a nice paper for Journal of Nothing Research, because it was just testing of a model that we had published in the American Naturalist, and it would just confirm something that mathematically we had already established. But we were there and it didn't work, and so on the spot we had to improvise, and we took a lateral direction and came up with something very original. Of course, it happens also a lot that it doesn't result in a paper in Science, but I still like this sort of, it's not the well-planned stuff that necessarily needs to go into these high-rating journals. Sometimes it's lateral thinking that gets you into something really new and really remarkable. Okay, and then the years spent. Any questions about this? Any remarks? Yes? No, they are points. So they don't, so yes, they can penetrate each other, but they are points, so we was just move on. So we didn't include bumping effects, no. That it would certainly be relevant. We studied it later, actually, and the bumping really changes their movement dynamics. They have a special way of moving. They do a levee walk. But if that means a sort of long-tail distribution, I don't want to go into too much detail. But if you then put lots of mussels there, they want to do a long-tail distribution and now and then take a big step, bonk into the next mussel. And so the whole levee walk becomes a Brownian walk when you have with increasing density of mussels. And in that context, you do see that it matters. At this stage, we didn't look at that. It certainly will also become, if you're in the middle of a clump, then you can't go anywhere, sure. Yeah, that's also an effect. But I think the guys on the border would still move out, and wouldn't that be a sort of delayed dissipation then? Maybe. Would it be interesting to investigate this further? Yeah. Okay. And then, one question. Yeah.
Just small question. What is the typical lifespan of a mussel compared to the time in which the pattern is formed?
That's a really interesting, difficult question. I would say 1 year, but the life, so every every spring, billions of larvae of mussels get born. From these billions, a few million end up on the tidal flat. You saw the pictures that I showed yesterday of these developing mussel beds. There were a huge amount of small little mussels there, making these clumps. A few weeks later, there were bigger mussels, same clumps, and it's still equal amount of open space. So lots of these small little mussels have died, and the few remaining are there. And then the average mussel bed, let's say 10% of the mussels beds survives the first winter. And so it's not like humans. We have a life expectancy and a life, but this is exponentially decreasing. And so what now the best value is, particularly if you start from the larvae, a day. But if you start from the clumps, the mussels that you see on the photographs there, I think they will most likely survive until the first winter storm, and then big chance of not surviving. Yeah.
Thank you.
And predation. Yeah. Yeah. So, well, you saw yesterday of course, I was terribly envious of Chris Klausmeier for his simple model and so on. And that paper And I know Chris, I met him a couple of times. The paper was really, I have been to conferences where there was a whole session, people analyzing the Klaus-Meyer model. So I was hoping now, because I had myself a paper in science, that would also happen with me. Well, yeah, forget it. So after a few years, I thought like, nobody's citing me. Why is that? And then I thought, thinking about this, I'm not really sure whether that's in any way true, but I thought like, well, I have this individual-based model. It compares to nothing. There are no comparable models like this, well, at least not that I know of. And there is, of course, the model by Klaus-Meyer, which nicely follows a partial differential equation paradigm. Is actually similar to the Gray-Scott model, which is also for other biological systems with a little bit of change. So I was sort of thinking, but did I make a good choice when I made an individual-based model? So I thought like, well, then we make a different model and see whether we can try again. So, yeah, Chris Klaus-Meyer's work. And so basically we had this basic idea that the movement rate of the organisms is dependent on the density of the organisms. And so this is something to build on. So how do you put it into a PDE model? Well, no idea. So, well, basically, of course, this is the general structure of an spatial ecological model, the Fisher model, basically. Well, as the most simple example. And well, if you want to change that, you have to make a density-dependent movement model. So the D, the diffusion rate, needs to change. So D must be a function. Of course, this is mathematically wrong. You have to do it like this. Then you have to put the D within the brackets. And so this is what that should look like then. And so then a PhD student of me, Chuanxing Liu, picked this up. He thought this was a really cool thing to study with. And so basically, what you then have to do, you have to translate data on individual movement into a population flux. And that was a bit of a difficulty. And it turns out, actually, what you're then doing is that you're doing the same as Albert Einstein did. Actually, Einstein's theory basically gives you the basis to make this translation from V, which is the movement rate of the individuals, to a population flux G, which, well, is the red line there. And so that actually they don't match much. The G should be the D. The equation and the plot don't really match here. And so what is striking, of course, is that although the V is always positive, the G dips through 0, becomes negative. So you have a sort of a switch from aggregation, so from dissipation, from dispersal, to aggregation. And when it really gets high again, you get again that there's again dispersal, but in between there's a zone with aggregation. And so we tried to make a model of this. And at first this, I have another slide, I think. Oh yeah, so if you want to simplify, I went a bit too fast. So the F here, of course, is not needed because I have a fixed amount of muscles. There's nothing happening. Of course, in the real world, now and then one dies, but just ignore that. And so basically we have sort of a mass conservation of muscles, or a muscle number conservation, that would be a better way to say it. And so, and then, yeah, Chuanxing tried to make a model, and it didn't work, because these muscles were aggregating and aggregating and aggregating and aggregating and more, and it went a single point with a density higher and higher and higher, basically a muscle black hole. And so, so we thought, like, what do we do with this? And then Chuanxing said, well, I will add a fourth order differential equation here. And when I looked at that moment, I didn't know about all these theories that you guys have learned about during your studies. I thought a fourth order differential equation with regard to space, it's like the derivative of the derivative of the derivative of the derivative. Well, I was lost. I didn't know. So I thought, oh, Chuanxing, if you think that's cool, then sure, do that. And so then he made a model. This is how it looks like. The same setup, grid, lots of variability to start with. And this is what it did. And so that's cool, I thought. Really nice pattern formation based on the individual-based model. So I thought, where did you get this fourth order differential, this fourth order term from then? And his answer was like, yeah, from the Kant-Hillard equation. And so, and that was for me, okay, my first introduction into phenomena of phase separation. And I found it really cool. Then I learned about phase separation, and you can just look it up at Wikipedia, of course. and about the patterns formation in alloys and all these things. So I found this was really cool and yeah, striking. So well, we did a whole analysis, and actually I must admit we didn't get much of the mathematics right. We went to mathematicians in Leiden, the group of Arjen Doelman, and he helped us to actually get our clumsy derivation following Einstein's papers. We did it all wrong. So he helped us get that right. And then we submitted it to a journal. Of course, first we went to Science, and hopefully that they would pull it off again. But they said, nah, we have enough of muscles. So we went to PNAS, and then we got the reviews back. And this reviewer was pretty critical, very critical, all sorts of remarks. And one thing was like, well, you know, I don't believe this. Unless this is the Lipschitz, when you follow the, well, I have a paper from this. The Lipschitz-Liosov law. And so the reviewer sitting there, Nigel. So at first, our response was, like, Lipschitz-Liosov law, come on. I mean, this is material sciences. This is like molecules and mathematics even. This is not real. It doesn't happen in muscles. Muscles follow, come on. So we resubmitted the paper. We dealt with a lot of the other comments, and I think we improved the paper. But we didn't follow this advice. So a few months back, we get to the reply back, and it's like, well, if you don't check on the Lipschitz-Liosov law, then I'm not going to believe it. Okay. So there we went. So first of all, we checked our model, and the model. I didn't explain the Lipschitz-Liosov law, but it basically was explained yesterday by Nigel. So basically, well, it just went a bit too fast, of course. The wavelength goes up as a function of time with a power of 1/3, as was explained yesterday. For me, the first time to hear about this. And so the model actually followed that pretty good. So the model agreed with that prediction. So that was actually something that already was quite remarkable. And then we looked at the data, and the data did this actually. So the first, let's say, 100 minutes, it indeed followed the Lipschitz-Liosov law. So we were quite surprised and very happy actually that Nigel pushed us into doing that. Because you can see three different densities, 1 kilogram, 1.6 kilogram, and 2.5 kilogram, and that one is a 2.5 kilogram. It follows it quite nice until about 100 minutes, but then it stops suddenly, like a breakpoint, like finish now. Why it's so abrupt, I have no idea. But then it flattens out to very. So that was something that actually I worried about. I worried that he would say like, oh no, doesn't agree because beyond 100 doesn't work. But actually, didn't happen. He was actually quite happy with it. So we got our paper in PNAS. And I think, yeah, I liked it very much myself, basically because I think we sort of came up with an alternative. Up to that point, if you would look at pattern formation in the ecosystem, they would all point to Turing's papers, the Turing principle for real-world pattern formation, not the spiral waves, of course. And of course, there's other examples, but the majority would point at Turing's activate inhibitor principle, which is growth-driven. You have a pattern, and in the places where you have the peaks, the growth of the organisms is just better than in the depressions. And we sort of came up with a movement-driven sort of self-organization in an ecosystem, and basically followed the phase separation principle. And there was so far not much mentioning of, at that stage actually didn't find any mentioning of phase separation in ecological or biological systems, although it turned out that in microbiology, the group of Michael Cates had done lots of things on phase separation. Okay, so yeah, here was the paper in PNAS, so yeah, I should emphasize the importance of Arjen here, Arjen Doelman. Basically, most of this, well, anyway, and we tried all sorts of things. We got it half right often, but he made sure that it was mathematically correct. Him and Vivi Rotscheffer, the two mathematicians in his team. Yeah, okay, so far so good. Any questions about this part?
So you mentioned the other day that the muscles, they adhere to each other, right? So they kind of have some sort of attraction in the sense that they really stay together for some time. So it's not only movement in the sense that they stay with a low speed around each other, they actually do adhere to each other?
Yeah, sure, this takes more time. If you go in the field, although it's a tidal system, so I go there and I go back, I don't stand there and monitor them, but you don't see that much movement, that they're fixed then, and also you actually, it takes quite some effort to dislodge them from each other. Although people describe that they can dislodge themselves, but it's a rather dangerous thing for a muscle to do. And so, yeah, in times it becomes really fixed. In the duration of my experiments, which is a day, it's, of course, they're better attached than at the beginning, and they make visual threads, but I'm quite sure that in the movies you always see them, they keep on moving a little bit. They keep on, it's not that they are completely fixed, although the coarsening stops. And that's, of course, a very interesting thing. And the coarsening is actually something, I just later on learned that the saturating effect in coarsening, so the arrested coarsening, is actually a bit of a topic here. And that's, I find really interesting. Also, because you would say, like, well, they go to a certain scale and then they stop coarsening. Well, they coarsen to 3 different scales, and it's on a log scale, so these are different scales, really. And of course, that deals with the density. So if you have less muscles, they apparently go further apart. They want to have a certain local density rather than a certain scale. So I think actually that there's lots of things to do here. If you want the data, go ahead. Although I think the group of, you're too late, the group of Nigel is, of Urban is already on top of that. So luckily he's not even listening. Anyway, yes?
I've got a quick question. Can you remind me, what is the difference between these different curves here? It's the total number of muscles?
Yeah, sure, the kilograms. So that's
So that's just, yeah.
that's the amount of muscles I tossed into the... So I wasn't going to... counting them sounds like a lot of work. So
Yeah.
I just weighed them, and that's the way
I don't remember if I suggested this in my review, which, but by the way, everything that Johan said is true, although I didn't say it quite as sharply as he had on his slides. I was a bit more diplomatic, and Johan didn't know it was me until after the paper was accepted. Then he wrote to Simon, right?
I think at some point when I learned that I had to take it seriously, I asked Simon, who is this person then?
Yeah.
And Simon asked you, Simon Levin.
Yeah, I said yes.
And then we actually communicated before we finished the paper.
That's right. But did you, since yesterday I talked about at the end about why in the biomolecular condensates they stop this T to the 1/3 and then go constant, and we had some discussion in there about the mathematical mechanism for it and various alternatives that we discussed with Owen and Pierre and so on. Do you have more data for different values of mass so we could see if it fits these? So in my paper from 1989, I'd worked out a scaling law that described this crossover. And did I ever suggest to you, if you have enough data, to test that scaling law?
I don't. The photographs is what I have as data.
Yeah.
So we did experiments. So
but
we did like this and like that.
You'd need many more mass. Do you have more data than those?
I think we might have even lower masses. Yeah. But it has been 20 years now. So or not. Yeah, this stuff.
10
actually,
years.
I did in 2006. Yeah, the paper is recently, but we first wrote the first paper, and so at that stage I wasn't doing that much with the muscles themselves anymore.
We should check. We should see if there's enough data to check that.
Yeah.
It might be relevant to what Owen did on biomolecular condensates,
like a very nice
or what Pierre was doing. So.
Well, but it's also the group of Ermin, one of his PhD students, who was contacting me about the saturation. So, definitely they are also involved. So maybe we should have a little discussion about this over. Yeah. Okay, to continue. So there's this. Oh, sorry. Go.
Your model doesn't show arrested coarsening.
Sorry?
Why your model doesn't show arrested coarsening?
I don't know. I can speculate, of course. I don't know. It should actually maybe do that because it goes to a certain pattern, but it may be also just a matter of time.
Okay.
I can give you.
In the model I was explaining yesterday, there's another physical effect that that may well be left out from his individual level model, and that might be that's why I'm asking my questions. But the most important thing is what does the data do, and then we can then try to figure out what is the left out physical effect.
Yeah, maybe to comment on this. I mean, if you have some kind of resources in your system, Johan, and these resources, here I am, and if these resources are produced and they're degraded, then you do have, from this resource production and degradation, you do have an effect which can be written as a Laplace law, a Poisson law, sorry, and then it's equivalent to a model where you have charges that interact in a long-range fashion. So any kind of chemical in the environment would do the similar job.
Yeah, well, there was surely, I think, a density-dependent effect on waste products that they have in the container.
Yeah.
We could flush it, of course,
Yeah.
although I do flush it already. But because you don't flush, of course, there are already lots of waste products. With regard to food, it sounds very logical. On the other hand, I didn't provide them with that much food. Of course, I had the photograph with the container, but that was just an early setup. So for the most, I fed them during the experiments, not sorry, in between the experiments, not during the experiments. So there's no difference in food availability here.
Yeah. So one would need to check several hypotheses.
Yeah.
Yeah.
Yeah, it would be nice. The whole setup has been disassembled, and so that would be really nice to do something about it. But that requires, well, time and space. Yeah.
So maybe just what I find, so they seem to be always coarsening for the same amount of time, which makes me believe whether it's related to some sort of, so for instance, in dictyostelium aggregation, they aggregate for a fixed amount of time, and then it doesn't matter, you just have more cells that are left behind after 8 hours. They have a very well-defined developmental program that is driven by starvation, how long a cell can starve, and then they stop aggregating, they stop signaling, that's it.
Yeah. I don't
and it seems
think
to
that
me, well, it's log scale, so it's hard to say, no? But
exactly. And I thought you see that the blue one actually levels off much earlier than the green one. The yellow one and is a bit fuzzy, but the blue one is really significantly earlier. It might be like half an hour. Although this is, I think each of them, I'm not really 100% sure, but I think each of them are individuals. So you need to we should repeat that actually to have actually a statistical measure of these things. Yeah.
So to address Ricardo's question, so I didn't talk about this yesterday, but there's another data collapse scaling theory that I'd worked out a long time ago, and that's the thing that we could see if these data fit that scaling theory. Yeah.
Okay.
Does these models reproduce the bigger patterns that you wanted to study initially?
No.
No?
No. Actually, I'm not really sure what to talk about Friday, but maybe I will. We did that. We made an integrated model. And it's a sort of weakly integrated model in terms of, we sort of impose it to scales, but we could see how they would affect the survival on average of the muscles and of the whole better in the end. So I might work on that to do that on Friday. Yeah. Okay. So I still have well, I will have half an hour left. So, well, phase separation in an ecosystem. Cool, isn't it? But yeah, how many examples? Like 1. So that's actually what we thought was a challenge. Can we find more of this back? And of course, a key thing that we had, apart from the movement that we have in this model, the other thing which is, of course, important is that you have mass conservation. And that's an important feature of much of the work of Kahn and Hilliard, of course, on phase separation in general. Typically, it happens under constraint of mass conservation. So we were thinking about, can we, as I explained already, I'm going too fast, I think. Well, yeah, here we are. And so can we think of other ecosystems that, well, not necessarily are having moving particles, but at least are driven by mass conservation. Although then, of course, there must also be movement, but not so much movement in terms of the, per se, as the animal movement, but that adhere to that. And so, well, could be. I mean, there's lots of ecosystems with patchiness, and you can imagine that there's some sort of a property there that sort of is a limiting factor. And, well, examples of that, this is a photograph of a peatland I showed you that also yesterday. And basically they are nutrient limited, and the most important nutrient there is phosphorus, and that's not going anywhere. Phosphorus are the input-output dynamics are much, much smaller than the stock of phosphorus in any ecosystem. Coral reefs, you don't know, but you can also see this little hummock there in the desert ecosystem. It's really absorbing sand underneath it, making a little hummock, which is then is the little dome, which of course then becomes a reservoir of water where the tree can grow in. And so, and of course here, although water is more of a flux system, the sand here is limited. There's a fixed amount of sand in this ecosystem. So we thought like, well, the mass constraint is actually something that you can find back in many ecosystems. Can we sort of argue that there is this principle is much broader applicable? And so we started looking at that, and it's maybe nice to give it a bit of a closer look. And you've already seen I'm a fan of quizzes. And so I want to make a little quiz here. This is a seagrass bed on a tidal flat. And the tidal flat is in basis rather sandy. Although underneath the seagrass, it's the green stuff, it becomes more muddy. And so would this be a system that would adhere to the principle of phase separation? And what would be then the constraint element in this ecosystem? Who has an idea? Anybody? So, what's an important resource here? Actually, the important determinant, to give you another suggestion, is that the seagrass grows much better when it's a bit muddy, because the mud contains nutrients, sand does not contain nutrients. Any idea? Well, it's the mud that actually can be a limiting resource. The sand, of course, also, but it's not that sand is concentrating under the seagrass bed. It's the mud that concentrates under the seagrass bed. Seagrass itself, of course, is not mass constrained because it's a living organism that just consumes water and consumes energy and all the things that it consumes. But the mud is just a limited amount of stuff that goes into the water, is trapped by the seagrass, stays for a little while, is then put into the water again. So it's basically a system where you have suspended sediment in the water, moving around when the tides go up. And there's this deposited sediment that sits underneath the seagrass, which basically exchange to become the whole body of sediment in this ecosystem. And what the seagrass does is actually mediate this balance between suspended mud and deposited mud. So this would be a nice example. And Koen was not necessarily interested. Koen is much more. Koen would feel perfectly at home in his audience here. He's much better mathematician than I am. And he didn't want to have like one example or make different models for all these examples. He wanted to make something that's sort of uniform. And well, we also worked on this one. This is, I already explained. Here, phosphorus is a limiting resource. So basically what you have mobile phosphates, which sit in the water, and you have phosphates in biomass. As soon as the plant takes up the phosphate, it becomes fixed phosphate for as long as the plant lives. And then if the plant dies, it becomes mobile phosphate again. So phosphate could be an important resource here. I've got also this picture. So it's actually a very nice example, I think. It's also nicely strange. So what you see here is a grass field, and the grass is, you see that there's sort of bimodal. You have these patches of really high grass, and the high grass is very spiny and, let's say, unpalatable. A cow doesn't like high grass. A cow likes short grass, because that's young, fresh. It's like cabbage or whatever salad stuff. So it's green, high nutrients. That's what it wants. So as a consequence, the cow maintains the short grass. I want that. That's what he first wants. If nothing is left of that, well, maybe he may think of eating something from the high grass, but preferably not. So you can already see that there's a sort of two states: a short grass state and a high grass state. But then, what would be a conserved mobile species here? Anybody? It's black and white. Cows. You put a certain amount of cows on this landscape. It's a farmer's landscape. So he just thinks like, this landscape can handle so many cows. Puts them on. If, of course, a good farmer would put so many cows on that everything is short grazed. But this is a nature reserve, so the density is lower. And what you get then is that some part of the landscape is not maintained by the cows because there are not enough cows to maintain everything. Other parts are maintained by the cows, but it's the cows that are the mobile, that are the conserved thing. And they basically, this is a picture of how the patch look like. It's nicely round, also very interesting. But to continue, so you have moving herbivores and you have grazing herbivores. Basically, a cow is either eating and grazing and consuming stuff, or it's moving and then looking for other grass to eat. And so when it finds a patch of short but not too short grass, it goes eating, and that reduces movement, shifts it into the grazing state. If it would encounter a high patch, then it would basically think like, well, nothing good here, move out. Let's go a little further. So you see that the movement of the herbivores is determined, of course, with lots of other things, by the quality of the grass. And so Koen wanted to make a model of this, and he also wanted to compare that. Koen was actually a PhD student of Max Rietkerk, working on arid systems and all these things. So he was used to making these sorts of models. And then you see that an arid system is a sort of a water flow-through system. So no mass conservation. The most important limiting resource flows through it. It goes from being surface water to soil water, and then, well, taken up by the plants and evaporated or flushed through. So it's a flush-through system. It's not conserving. Of course, if you look at these other, I'm going a bit too fast. For the models that he had been thinking of these three example ecosystems, you have to make a different model that doesn't no flow through in there. And so he started to work with Arjan Doelman and Vivi Rotteschaffer again. And basically, we made an outline that you have been perfectly explained to you by you yesterday. Well, in general terms, you have two elements. You have fixed particles and mobile particles. There's both are diffusing with different rates, and you have functions that determine the reaction terms of both the U, the fixed particle, and the mobile particle has a reaction term G. And of course, there's mass conservation. So basically, I think the phosphates is the most clear example. You have phosphates in the water. It gets taken up by the plants. It becomes then a certain amount of biomass with a concentration of phosphorus, but no phosphorus gets lost. So basically, if we would make a conversion, then G equals F minus times the conversion C. And so that means that this one is just, well, they're basically just equal to each other, but have a negative sign. One has a negative sign, the other has a positive sign. Taking into account the conversion. And of course, there's global mass conservation, as is said here. And so this is the basic model that we did. And he made a sort of a minimized model of that, which I think, I pressed the wrong button. So this is the F, this is the G, as I explained. And this is the model that he in the end made. So he converted the model in more general terms. This have an F and a minus F. That was also explained yesterday by I'm not sure if Nigel or Erwin. And we used this specific function of F, which Koen derived. So we made a model for all three ecosystems, and arguing from that, we derived to this general equation, and so to say. And so this is our generalized equation for phase separation in ecosystems. And of course you have global mass conservation. And if you run this model, it does like whatever all these models do, and they look exactly the same. So you get this phase separation model. And Koen was then starting to analyze this, looking at the dynamics. And of course you could already see here in this model, you see that some of these patches are growing, other patches are disappearing. And you can analyze this, and then you see that well, the green ones are the ones that are growing, the red ones are the ones that are disappearing, and in the end, of course, I mean, gradually the ones in the green will cross the red border and drop down, and you end up with one big patch left over. And so, and that depends on the critical patch size, basically. So as small patches disappear, the average patch size changes, so meaning that the next smallest one is the one to suffer, and you have a critical patch size which actually changes with the average patch size of the whole system, and it divides the expanding and the declining patches with regard to their patch size. And what is actually interesting, if you would look for the patch size distribution of an arid system, so a Turing system, you would see that they would, given the right conditions with not too much resources, they would develop to a characteristic patch size, as you can see here. And this is a bit of a fuzzy curve, of course, but what we do is we basically look at all the size of the patches that are basically equal or larger than the value what we have on the r-axis. So that means that that here they're all included, and as you move through the patch radiuses, and suddenly you go to the patches the size that you see here, and then you move down until you end up here where none of the patches has a size of this scale. If you look at the phase separation examples, what you see is that, of course, roughly it develops to a similar shape, but this shape is constantly moving. And because these patches are increasing and increasing and increasing in size until you have basically one patch left. However, if you look at this patch size distribution and you divide it by the average of the patch sizes, they all merge to the same patch size again, to the same distribution again. So there is a characteristic distribution, only it's relative to the average patch size. And so one thing that I sort of put out, which is the funny ecologist approach of analyzing a model, you just do a simulation. So we wanted to have two ecosystems. One is a Turing ecosystem, and the other is a phase separating ecosystem. And there is a disturbance in this ecosystem. Part of this ecosystem, the vegetation disappears. And of course, important is, does it grow back or not? Well, if you do that with the, well, here you can see them both. This is a gap in an arid vegetation. This is a gap in a density-dependent aggregation system, so a phase separating system. And so if you then let these models recover, then you see that the Turing system just repairs itself, goes roughly back to the same situation. The phase separating system doesn't do that. What you have here is that the gap, well, it's sort of roughly still there, but the resources or the nutrients or sediment that was captured by the plants in here disappears to the other patches. So on a larger scale, you see actually that the cover is maintained. There's still mass conservation, but locally, it's not resilient. And you might think like, well, that's a, sorry?
In the left system, is this a Turing system in the sense that there is no mass conservation?
That's just the arid systems model by Max Riekerk. So that basically the water flow. I showed it on the yesterday. Because you have this three-component model where you have surface water becoming soil water, becoming plants, and then out. So water is not conserved.
Okay, and then locally you can tap into a reservoir and thereby regain the pattern.
Well, if
on the right-hand side, you have to have mass flow, which is way slower, right? Isn't that the argument?
You have there
If
is
mass is
fluxes.
conserved,
There's fluxes.
then you have to flow.
Yeah. Yeah. Sure.
And you cannot tap into a reservoir
But it's
to
also
regain the equilibrium.
it's also captured by the patches surrounding. They still have that biomass, so it doesn't flow back. It's captured by the other patches, and they grow, and they're all competing for the resources.
Yeah.
And so that this is sort of an ecologist terminology, but it's sort of they're all little beds of seagrass, whatever you want to make them. As soon as one patch is hit, the others are happy to extract the resources. They are released into the yellow space, orange space, and then captured by the other organisms. That's the idea. Yeah. Which, of course, if you think about it, it's quite important, because if humans made a little hampered with some ecosystem, you burn the forest, to sort of put it into a Brazilian perspective, and so you can repair the Turing system. You can help it maybe, or it repairs itself. It's repairing. You can, so if you would want to facilitate that as a manager, you would plant plants in the Turing system, you would actually facilitate it recovering. But if you do that in the seagrass system, which this as an example, and actually we tried that without knowing these dynamics, to sort of help seagrasses recover, they all die. And that's, and maybe even on the larger scale, other seagrass patches are expanding, but locally you cannot repair that. And that's for a manager important knowledge. And so it's a fundamental difference in how you deal with these ecosystems. You can locally try to restore a Turing system. You cannot locally easily try to restore a phase separating system. Okay, and so if you look at the explanation behind this is basically how patch size relates to growth rate. In a Turing system, there is a negative relation between patch size and growth rate. Basically meaning small patches have an advantage, large patches have a disadvantage. And you basically thereby end up with a sort of typical patch size, because any smaller patch size starts growing until you end up here in the middle. With a density-dependent aggregation or phase-separating system, we of course put an ecological label on top of it, you have sort of the opposite relation. You can already see that it's unstable. That meaning that if you are a bit bigger, then you start growing, you can become even bigger. If you're smaller, then you start growing and become even smaller. Of course, this line here is not a fixed point. It's a line that is constantly moving as the patches here are disappearing. I mean, that also means that if you disturb a big patch and you push it over the boundary, it disappears also. And so this is the way that we explained basic differences in the dynamics of these two types of systems.
Sorry.
So I'm just confused about a very simple point. So the scale-dependent feedback, that's the Turing system, right? And the density-dependent aggregation, that's meant to be the Cahn-Hilliard.
Yeah. I should have used different labels in this audience.
But when you looked at the, what I'm confused by is when I looked at the pictures on the previous slide for the arid vegetation, this one. So this looks like a Cahn-Hilliard pattern, right?
That one.
The left one, yes.
It isn't.
But it's not.
But it's because it looks like it because it looks like it's yellow and then suddenly green. It looks like two patches, but that's the result of because it's a three-link system. So if you look at Klaus-Meyer's model, it's much more wavy, and this model, because it's a three-link system, has tendency to be less wavy, more like two states.
So this has a conservation.
No, this is a flow-through system. It's water.
So no conservation.
No conservation.
So the one on the right has the conservation.
This is conservation. That is not conservation.
Yeah, okay.
I should have labeled it a little different given his audience.
Yeah, thanks.
Yeah. Okay.
So maybe Johan, one quick question from my side. What happens? So if you have different environmental conditions, so worse environmental conditions in the sense that the front in the Turing pattern would be desertifications instead of recovering. So you are in the other side. You are past the Maxwell point in the propagation of the front.
That's an interesting
do you change? Do you have the different, like the Turing is weaker than the?
I have not tried that, so it's difficult for me to say this is it. But it would be interesting indeed for both. I think the dynamics of that thing will not differ. You will just have less cover And because there is no tipping
That,
point,
that is my question. Yeah.
and that's you have to stay to well you have the spinodal region and so it might be interesting to see whether there's fundamental differences in the spinodal region and in the other what was
Nucleation.
the other region.
Nucleation.
Sorry.
Nucleation, no? Nucleation.
Yeah, yeah.
But that's there, no?
The right one is the Carn-Hilliard system, but in the nucleation regime. That's right. That's what you're saying.
Yeah, that
That's
might
what I
actually be an interesting point. That
Right,
the
and
regimes
so-
are not. So we should actually try that with a lower
That is-
biomass and
yes.
see whether. and
Exactly.
I'm actually quite convinced as you approach the tipping point, you might get a runaway response there. That you disturb it and then.
That was
Yeah,
my...
yeah.
And whether you can calculate the Maxwell point, no? There in the...
Yeah,
because
yeah,
that's
yeah,
a
yeah,
front
yeah,
at the end.
exactly. But the one on the left, you say that one is resilient or not resilient?
In this simulation, that one is resilient.
Well, it's resilient because something like the original pattern grows back.
Yeah, but I would say it's locally resilient because
Well,
that
that's-
one is also resilient.
Right, they're resilient in different ways. One is local, one is
Global.
right. I mean, the droplets sort of respond in a different way,
Yeah.
and you still see the remnant of this square perturbation at long times. If you did it in the spinodal regime, it might be different, right? It'd be faster.
Well, but you still also have the same phenomenon that the resources that were captured in that little are released in that disturbed area will move in the other patches.
Yeah, everything's
I
moving
think,
around. Yeah.
I think it might differ, but it might still give you the same result. But it's also well possible that the other ones are then becoming so big they will just grow into it more. And this is a more so you can see that the cover is not really comparable. It's actually a good point.
But I think it's an apples to. I think we're all saying the same thing.
Yeah,
It's
I
kind
agree
of
with
like
that.
an
Yeah.
apples to oranges comparison, as it were. It'd be interesting to try it in a different regime.
I should try that with a lower biomass, and you can of course try that for the whole regime of rainfall or nutrients or resources, and see how that fundamentally changes. Then and you have the whole picture. Yeah,
Yeah.
lots of stuff to do.
Very interesting.
Yeah. Okay, I think this finishes my talk. So nice paper again, and well, we hope that this idea
Johan.
gets going on. Sorry.
Don't worry. I am curious about in the previous slide. What are no the next. What are the points? What are your data points there?
The patches that you see.
You measure in this ecosystems.
Of course, we then used it's models.
Okay.
No,
okay,
it's all models. This is this is the typical theoretician's approach among the ecologists. We go take a few photographs and then we sit behind our computer. It would actually be nice because actually the seagrass bed, this photograph is. Come on. This is 200 meters, 300 meters from my office door, so maybe a bit more, maybe a kilometer. So, yeah, it's open for experimentation. Yeah. Although I'm quite sure that seagrass is not doing very well in the area. I might need to have to do some convincing with the local managers. Particularly if the predictions is that if you perturb it, it will not recover. I will not be very convincing here with my models in my hand. But yeah, on the other hand, we should think about something. Yeah. Okay. Yeah. So far, so good. I'm finished. Any further questions? I had some nice last slides which I could maybe show.
So maybe before the last slide, Johan, there is a question here?
Yeah, sure.
Or what?
So thank you, Johan. I believe the simulation on the right, if you change parameters, in the spirit of what we are discussing here, you would get a bicontinuous structure as well, right? Because the left one has this bicontinuous kind of pattern, whereas the one on the right has droplets. If you change the parameters, the right one, the phase separation one, would also have this bicontinuous structure. And if both of them have bicontinuous structure, would these be sufficient to give the same resilience for them? Or
Well, it's of course comparing apples and pears, but whether the prediction that, here we predict that local resilience is different from global resilience, and that the Turing pattern is not locally resilient, and the other is. I'm quite sure that it will be dependent on conditions, so it's not a universal prediction, but whether it will completely fall down, yeah, I don't know either. Should do that, yeah. Nice. We have 1.5 hours tomorrow. Can we do this? Yes. Anyway, yeah, to finish, I want to show my last slide. Thank you for your attention. At the 1st of January, a PhD student is starting in this system. It's a study on the resilience of the water sea to climate change. And of course, for this system to follow climate change, nutrients and sediment need to come in. And so the plan, there's a whole study, many people, physicists that are studying water flow, sediment transport, the whole lot. I am tasked to look at the interaction between sedimentation and the biota. And so basically I want to put the whole idea of this local resilience, global resilience and so on in this ecosystem where you have these creeks also. And they are sort of highways of, so imagine yourself making a model of a phase separating system, but you put little zones in there that are highways where the sediment can suddenly go much faster. And so see how that affects the, in both ways, how that affects the dynamics of these of the resources, and also with regard to the resilience if the people disturb this. And they want to make disturbance by means of dumping sediment in the landscape to help it recover. On the other hand, I think also the phase separating system will influence the creek formation. And so actually I think there's lots of interesting things to investigate, and a new PhD student will start in the 1st of January. Would be great for her to be here, but it was too difficult. Okay. Anybody? Thank you.